Hi. So, in previous session we have learnt about the TLR, toll-like receptor. We have learnt about the various ligand. We have learnt about the signaling pathway, and we have also learnt about how the mutation or single nucleotide polymorphism or single nucleotide variants result to the development of some or other disease, and in some cases it plays an important role in protection against disease. And we have also discussed in a great length that there are some molecules which the researcher made, and these molecules are acting as a agonist or antagonist, okay, and these molecules are used in treatment of cancer, okay, and they are also used in bacterial and viral infection, and they are also used for autoimmune disease, asthma, so and so, okay. So, in this session we will discuss about one very important pattern recognition receptor which is expressing in the cytoplasm of cell, okay. So, the basic question is when TLR was already discovered, then there is a few basic questions were there in the field. As you may aware that most of virus, they replicate in the uh, in the cell, they use the host cellular machinery, and there are some RNA species are present in the cytoplasm, okay. And these RNA species which is present in the cytoplasm, they are basically, there must be some sensor which can sense this these RNA molecules in order to elicitate or activate or induce appropriate innate immune response, okay. So, this was a basic rationale in the field when lot of TLR work was going on, okay. So, there was a kind of need that, so how these species of RNA are sensed, and there is development of appropriate innate immunity. So, when I say innate immunity and virus infection, then one obvious response is the type 1 interferon production, okay. So, this is a quite obvious. So, the RNA species is present in the various RNA species present in the cytoplasm, and how this is inducing a response, okay, innate immune response or induction of type 1 interferon. So, there was a discovery of some molecule before discovery of RLR. So, these molecules are protein kinase, okay. If you remember, there are some interferon inducible gene, okay. These interferon inducible gene, one is protein kinase, okay, and another is, if you remember, there is 2' 5' oligoadenylate synthase enzyme is there, okay. This molecule which is playing important role in antiviral immunity, okay. And this 2' 5' oligoadenylate synthase, basically their end product activates RNase L, and that checks the viral replication, okay. However, these molecules are not able to induce type 1 interferon, okay. So, that was the basis for the discovery of RLR or cytosolic sensor. In 2004, group of Takeshi Fujita in Japan, he extensively performed a screening experiment, and he found out that there is some molecule, okay, which can sense this RNA molecule, and this molecule can induce the synthesis of type 1 interferon. This is the original paper. If you see this paper, this was published in 2004, okay, in Nature Immunology, and this was the first report or discovery paper for the identification and characterization of cytosolic sensor, that is RIG-I. Okay. And after that, this field expanded a lot. There was a discovery of other members of RLR family protein, and so on. So there was a discovery of the adapter molecule for this RLR sensors was taken place, and then all signaling pathway was dissected out, and then this field was very well established. Okay. Now I will tell what are the members of RLR family sensors or PRRs. So here you can see there are RIG-I, which I have discussed. The RIG-I is basically the retionic acid induced gene 1, and if you see this protein, this has a card. There are two domain which we call it as a card. So card is basically stand for, there are I have seen the literature, people has a two kind of full form. One is caspase recruitment domain, which is originally given in Takeshi Fujita's paper, okay. And in recent several articles, it is also written that caspase activation and recruitment domain, okay. So don't worry about that. You should know that there are two similar kind of full form of card, okay. And this, as the name suggests, RIG-I, it's a retinoic acid induced gene, okay. Its another name is DDX58, okay. The another member is MDA5. MDA5 stand for melanoma differentiation associated protein 5, and this is also known as IFIH1, and this protein also has a 2 card domain, and there is a RNA helicase domain, okay, in both cases RIG-I and MDA5. Here it is shown in purple color, okay. So this RNA helicase domain is very much essential in order to bind with the ligand. You will see I have a schematic for how this ligand, the RNA molecule, is binding with this sensor. So these are two major sensors, okay, and there is one more sensor which we call it as a LGP2, okay. LGP2 is, it's a little different name. If you see, it's a Laboratory of Genetics and Physiology 2, okay, the name of the gene, okay. So this gene or this protein doesn't have a card, okay. They have only helicase domain or RNA helicase domain, and there is a CTD, it's a carboxy terminal domain, okay. So towards N terminal there is a card domain in case of RIG-I and MDA5, and there is a towards C terminal there is a RNA helicase domain in both RIG-I and MDF5, and there is a carboxy terminal domain is there, and this domain is also playing important role in sensing. Okay. Very interestingly, this LGP2 doesn't have a card. Okay, the card domain is not there. This has only RNA helicase and CTD, carboxy terminal domain. So when this molecule was identified, people thought that this molecule is a kind of a negative regulator of RLR pathway signaling. Okay, because this can bind with the ligand, but this cannot transduce the signal. Okay. Please note, the card domain is needed for the activation of downstream signaling. Okay. So, since this LGP2 does not have a card, so people thought that this is maybe a negative regulator. Later on, the in vitro studies suggested a similar observation that this molecule is a kind of negative regulator, but genetic studies means the knockout mice created for the LGP2 does not show that phenotype. Okay. So, this is little debatable that LGP2 is a positive modulator or positive or it is a sensor or it is a negative regulator. Okay. So, how this or what are the ligands recognized by RIG-I and MDA5? So, originally there are several viruses are reported to recognize in these studies like Dr. Fujita's studies and subsequent studies, they extensively use these viruses. Okay. So, RIG-I basically recognizes if you see in this slide, there is a Newcastle disease virus. Okay. So, Newcastle disease virus is not pathogenic to the human, they infect the birds. Okay. And this virus we can easily use it in the lab as they are not affecting the human. Okay. So, NDV is a one virus, another is vesicular stomatitis virus, VSV. You are probably very well aware about the VSV. Okay. And there is a Sendai virus. Sendai virus is also not pathogenic to the normal human. Okay. So, these viruses we use it for the study of RLR or RIG-I pathway. Okay. In addition, this RIG-I also recognizes a chemically synthesized ligand, that is 5' triphosphate. Okay. The RNA molecule having this 5' triphosphate. Later on it was shown that in addition to 5' triphosphate, they also recognize as diphosphate or to some extent monophosphate as well. Okay. So, these are the ligands for RIG-I, and for MDA5 people used this EMCV virus, encephalomyocarditis virus. Okay. And in addition to EMCV, this also recognizes a chemically synthesized molecule which we call it as a poly-IC. Okay. Poly-IC is a polymer of inosine and cytidine. Okay. And when this polymer is introduced inside the cell, then this mimic like a virus infection. So, this poly-IC is extensively used in this field. Okay, in RLR signal, in investigation of RLR signaling pathway. Okay. So, there are a variety of poly-IC, low molecular weight, high molecular weight, and it is considered that the MID88 recognizes a high molecular weight poly-IC. A low molecular weight or short stretches of poly-IC is also recognized by RIG-I. Okay. So, after that this once you introduce this these ligands, then this RIG-I and MDA5 pathway activates, and then it induces type 1 interferon and pro-inflammatory cytokines. Okay. So, I will talk more about the signaling in next session, and over there we will discuss what are the signaling pathway, how this type 1 interferon as well as inflammatory cytokines are induced in the cell. Okay. So, this is a very interesting schematic. Here you can see that this RNA molecule which is having the triphosphate is binding with the RIG-I. Please note, RIG-I is extensively studied, it is a crystal structure and everything, okay, post translational modification, so and so. Okay. So, this is a schematic representation that how the RNA molecule is sensed by the RIG-I, means how RNA molecule bind with this RIG-I. Here you can see there is a CTD, this is carboxyl terminal domain. This is basically binding with this triphosphate, it is holding the triphosphate, and it could be a diphosphate also. Okay. And here you can see the ribose sugar and there are bases. Okay. In RNA also there is a you remember the structure of RNA. Okay. So, that has a nitrogenous basis and this is a ribose sugar. So, basically this is recognized or it is binding with the helicase domain, and it is considered that this when there is no ligand, then this CTD mask this helicase domain and stop the activation of RLR signaling pathway. And once ligand approaches to the RIG-I in the inside the cell, then this helicase domain is basically unmasked by the ligand, and then this is binding with CTD and helicase domain. The ligand is not binding with card of this protein. Okay. This is this domain, it is not binding with card. Okay. So, card is basically involved in activation of downstream signaling. Okay. So, now I will discuss about this what are the various viruses which is recognized by this RIG-I and or MyD88. Okay. Here you can see this is a Baltimore classification. Probably you might be aware that what is Baltimore classification. It is basically a type of classification system which places viruses into one of 7 classes. Here you can see there are 7 classes. Okay. 1, 2, 3, 4, 5, 6, 7. Okay. And or group 7 classes or group based on combination of its nucleic acid, that is nucleic acid could be a RNA molecule, it could be a DNA molecule, and the strand they possess. For example, there is a double stranded DNA, there is a single stranded DNA, double stranded RNA, single stranded RNA with positive sense, okay, single stranded RNA negative sense, and so on. So, okay. So, this is the basis of this Baltimore classification. So, as you can see in this Baltimore classification, so RIG-I and MDA5 can recognize all kinds of viruses, except if you see there is this RLR sensors, they do not recognize, or it is not known at the moment that they recognizes some single stranded DNA viruses. Okay. So, we are not aware. Let me take you with each class. So, first is that double stranded DNA. This is basically in this class there is a herpes viridae family viruses are there. Okay. This is basically consists of herpes simplex virus, okay, which cause a Kaposi sarcoma, and in the same class there is a Epstein-Barr virus, okay, which if you remember, or if you have studied, this cause the Burkitt lymphoma, okay. So these viruses are recognized by RIG-I and MDA5. They also recognizes pox viridae family member, which is vaccinia virus, and this is also recognized by both RIG-I and MDA5. Adenoviruses are basically recognized by only RIG-I, okay. So this is a class 1. Class 2, single stranded DNA, it is not known that any of this RLR member recognizes single stranded DNA molecule. The third class is double stranded RNA viruses, which is basically Rio viridae family members, and the best example is rotavirus, and this is recognized by both RIG-I and MDA5. The fourth class is single stranded RNA, which is positive sense RNA viruses, which basically consist of Picorna viridae family viruses, and the best example is EMCV. You have seen in previous slide, okay. So EMCV is recognized by MDA5, but this is also recognized by RIG-I. The rhinoviruses are also recognized by RIG-I and MDA5. Coxsackie B virus is also recognized by RIG-I and MDA5. Flaviviruses, which is again the class 4 single stranded positive sense RNA viruses, and this is a quite in disease point of view, this family, the Flaviviridae family, is quite dangerous if you see carefully, okay. You know that all these viruses like West Nile virus, hepatitis C virus, Zika virus, so all are quite dangerous or pathogenic, right, and they are recognized by RIG-I and MDA5. And your the most important virus, probably all of you are extremely well aware about this virus, that is coronavirus. So this coronavirus is which belongs to the coronaviridae family member. Coronavirus is basically SARS-CoV-2 or CoV-1, okay. So this virus is sensed by both RIG-I and MDA5, and there is a class 5 single stranded RNA negative sense RNA viruses, and the most again fatal virus is orthomyxiviridae family member, and in that one of the key member and very famous member is influenza virus, okay. So this influenza virus is exclusively recognized by RIG-I, okay. There is a paramyxoviridae, which is a family member, and this virus is basically a measles virus, okay, and this is also recognized by both RIG-I and MDA5. Filoviridae family member, which is consist of again very dangerous virus, that is Ebola virus. You probably know that Ebola is causing a massive hemorrhage, okay, and people die in very short duration, okay. So Ebola virus and Marburg is also equally fatal. This is also recognized by RIG-I and MDA5, okay. Single stranded RNA with reverse transcriptase activity. So this is retroviridae. The best example is HIV. This is also recognized by RIG-I and MDA5. Double stranded DNA virus. This is Hependiviridae, which is basically hepatitis B virus, is also recognized by RIG-I and MDA5. So here you can see most of the viruses, it's irrespective to the DNA or RNA, they are recognized by this RIG-I and MDA5, the key member of RLR family protein, okay. Now I will discuss about it's not RIG-I and MDA5 is not only sensing this viral origin nucleic acid, this can be activated by host RNA, and this host RNA, which is sensed by RIG-I or MDA5 is activated during the infection. Okay. Again, I will take your attention to this fact that when the virus infect the cells, okay, at that time they basically make their own copy. And how they make their own copy? By hijacking the host cellular machinery, right? They hijack the host cellular machinery and then they use this machinery to make their own copies, either nucleic acid or protein, and finally they assemble, and then there will be aggression of virus or release of virus. And this newly found virus or progeny of virus, they again infect another healthy cells. Okay. So this is a very simple life cycle of a virus. Okay. Try to understand, when they hijack, then these cells are under stress, the host cells are under stress. Okay. And this stress can also result to the dysregulation of particularly, if you see carefully, the nucleic acid metabolism will be quite severely dysregulated. Okay. And over there is a possibility that our sensors, which is basically expressing for protection, may start recognizing our own RNA molecule or nucleic acid, and then that will cause the problem. So here you can see that after herpes simplex virus infection, okay, people found out that this RIG-I is strongly associated with our own RNA molecule. Okay, and this RNA molecule is RNA 5sp141. Okay, this is basically a 5s ribosomal RNA, and this 5s ribosomal RNA is basically a pseudo gene transcript. Okay. So you can understand, due to this virus infection, this RIG-I started binding or recognizing the host 5s RNA. Okay, and that will cause activation of signaling. So when this is happening, then there will be a mislocalization of this RNA unusual, which is unusual. In normal scenario, this is not happening, the mislocalization or misprocessing. Okay. So this infection basically result to this mislocalization and misprocessed. Overall, if you see very carefully, there is a disturbance of this nucleic acid metabolism or nucleic acid homeostasis. Particularly in this case, the RNA homeostasis is severely skewed. Okay, and this skewed process basically result to the binding of RIG-I with our own RNA molecule. Okay, that is this infection also cause unmasking of non-coding RNA. And so probably you might be aware that now there are various species of RNA molecule in the cells. Okay, and all these species of RNA molecule basically play a very important role in gene regulation. Okay, they are kind of fine tuner and they act at transcription level. Okay, so this fine tuning is basically caused by a variety of RNA species, which we call it as a non-coding RNA. This is a quite chunk of RNA. Okay, non-coding RNA is a quite a chunk. If you see the genome, only 98, so only 2% of genome makes protein, and rest of genome we were not aware, but now we are having evidences that they encode for various species of RNA. So if you see this various species of RNA, they are not just expressing like that. They play a very important role in transcriptional regulation. Okay, and this transcriptional regulation is basically taken by various non-coding RNA. For example, there are long non-coding RNA, there are short non-coding RNA, there are circular RNA. Okay, so all this plays a very important role in regulation, and among this short non-coding RNA, one microRNAs are also there. So microRNAs these days, there is a huge evidences of this microRNA play a very crucial role in regulation of any gene. Okay. So basically here you can see the cell is loaded with a variety of RNA, and if there is some disturbance in homeostasis of RNA metabolism, okay, then that may result to the activation of RLR pathway. So this in previous slide you have seen there is some disturbance in cellular RNA homeostasis by virus infection. Now there is also evidences that this RIG-I can get activated by itself, and without any perturbation. Without any perturbation means without any virus infection. Okay, and if you this is quite obvious and common that there must be some individual who is having some mutation in the members of RLR protein. Okay, so mutation in RLR cause the spontaneous signaling in absence of any virus infection. For example, if there is some mutation in CTD, okay, which will keep it in unmasked form of helicase, then probably that will keep on activating. Okay, and that is a point of concern. So there are two diseases which is known as Singleton-Meriten syndrome and Aicardi-Goutieres syndrome. So these two syndromes are basically in these individuals there is a mutation in the member of RLR pathway protein. Okay, or sorry, RIG-I or MDA5 sensors. Okay, so in case of this Aicardi syndrome, I am speaking for convenience because the long name is little complicated. In case of Aicardi syndrome, there is a long hairpin RNA derived from inverted repeat. There are ALU element. Okay, with a lot of ALU element. So repeated ALU element have been suggested to activate mutant form of MDA5. Okay, so this mutant form of MDA5 basically activated by this inverted repeat of ALU element, and that will cause activation of RLR pathway. Alteration in the RNA metabolism can generate the RLR stimulatory RNA. Okay, so if there is some alteration in RNA metabolism, for example, some enzyme lacking or expressing more, so that can generate the stimulatory RNA, and that stimulatory RNA may activate this RLR pathway. Okay, there could be a possibility that there is a mislocalization of mitochondrial RNA. Okay, into cytoplasm, and that may lead to the MDA5 activation. Okay, so there are some more situation in which there is activation of RLR signaling pathway, that is adenosine deaminase acting on RNA1 deficiency. This if there is this enzyme deficiency will be there in human or mice, that result to the chronic type 1 interferon production and inflammatory, and that chronic production of type 1 interferon result to the inflammatory pathology. Okay, there is evidences that in breast cancer the tumor microenvironment result in increased production of non-coding 5' triphosphate containing RNA, that is RN7SL1 in stromal cell, and that followed by transfer of this RNA via exosome. So, exosomes are a vesicle which is released from the cells, and this exosome is loaded with a variety of molecule, and basically these exosome, the molecules which is present in the exosome, they tell about the health of cell or eventually the health of host. Okay. So, in many scenarios these exosomes are used for future diagnostic. Okay. So, transfer of this RNA via exosomes to cancer cell, and that result in activation of RLR pathway, particularly RIG-I. Okay. So, now in this session you have learnt how the RIG-I pathway was discovered, okay, or how RIG-I is discovered, and what are the various ligand for this RIG-I and MDA5, okay. And here you also learned that this RIG-I or RLR pathway is not only playing or getting activated during virus infection, it is also activated by host. Various RNA species can activate this RLR pathway. Okay. In this field, the RIG-I is quite extensively studied, but MDA5 was not that extensively studied. So, this is just for your note. Okay. So, with this I will stop this session, and in next session I will discuss about the RLR signaling pathway, what are the signaling things are there, and I will also discuss this RIG-I and MDA5. They are undergoing lot of post translational modification, or there is a microRNA mediated regulation. So, I will just show you the complexity, that how this RIG-I and MDA5 how complex the regulation of these proteins are there. Okay. And finally, I will discuss about the disease associated with RLR signaling pathway, and there are some molecules which is used for used as a therapeutic in order to activate the RLR pathway, and there are some molecules which damp this RLR pathway. Thank you. We will see you in next session.